Okay, we're back with Chapter 6. Today we're going to focus on cell structures, and specifically, in this video, we'll take a look at, the nucleus and ribosomes. So let's take a look at their structures and what they do, their jobs. This slide has descriptions, what we want to write down, and the next slide has a picture of the nucleus. Now, we're taking a look at cell structures that are found in eukaryotic cells, not prokaryotes. So eukaryotes have these membrane-bound organelles. An important one is the nucleus. The nucleus is a membrane-bound organelle, and it actually is made up of two membranes. So let's write that in there. What that means is it has a double membrane around the nucleus. Remember that a membrane is made up of phospholipids. Actually, a membrane is made up of a phospholipid bilayer, if you remember. Each phospholipid points its head out and tail inward. That's one membrane that I've drawn. A nucleus has two membranes, so we'd have to draw a second membrane. I don't have much room here for my drawing. Okay. There we go. Two membranes in a row. And it's found in eukaryotic cells. Cells that make up animals and plants and fungi and protists. It's big relative to the size of a cell. Cells in the eukaryotic world are 10 to 100 micrometers in size. A nucleus can be as big as 5 micrometers in size in terms of its diameter. So when we look at the nucleus, let's think about the nuclear envelope first, and then we'll look at what's inside and kind of what happens inside. Let's take a look on the next slide, because the next slide has some nice pictures. These are found in your text. So the nuclear envelope is actually made, so here's a nice description right there, is made up of an inner membrane, one membrane of a phospholipid bilayer, and an outer membrane of a phospholipid bilayer. And then there are hundreds or thousands of pores, nuclear pores, that run through the membrane so that materials can pass from the nucleus into the cell, or from the cell and into the nucleus. So when we draw a membrane, there are going to be these pores that allow materials to pass across the double membrane. We have this double membrane that I'm drawing in here. Okay? These nuclear pores are made up of a set of proteins, and there are 8 proteins that kind of ring each pore. The pores are very small. They're 100 nanometers in size. And, they look like a little flower almost. And they allow materials to go into the cell or out of the cell, but they're very selective. Think of them as kind of border guards, these pores. Only some things can pass into the nucleus. Things that the nucleus might need, ribosomal proteins or nucleotides or energy. And some things pass out of the nucleus, like ribosomal, or like RNA, for example. So these are very selective, and that's important. The nucleus holds the genetic material. So here's where we are. Let's see. Here's a nice picture of these pore complexes made up of pores that make up the pores. Inside, and I forgot, inside the inner layer of the nuclear envelope is a nuclear lamina. It looks like this nuclear lamina is made up of kind of strong ropes that are kind of crisscrossed. That's to, we think, provide structure for the nuclear envelope and for the nucleus as a whole, to give it support and structure. Inside the nucleus is the genetic material. We call that genetic material the chromatin. Chromatin is made up in eukaryotes of many strings of DNA and proteins, but that has the genetic material. The DNA holds the information, the hereditary information for the cell, so that the cell can act and behave the way it does, whether it's a liver cell or a neuron or a muscle cell inside of us, or whether it's, a cell that's important for a leaf to make, sugar. Okay, the DNA inside that's a part of the chromatin is the genetic material. Inside humans, we have 46 strands of chromatin. Each strand is different length and carries different numbers of genes. And these chromatin strands consolidate or coil up to form chromosomes during some part of the cell's life cycle. And if you have an unusual number of chromosomes, that's usually not good for the organism. Okay, and a couple other things about what happens in the nucleus. So we've got the important genetic material, but the genetic material is in fact copied. Pieces of the genetic material are copied in the nucleus to make RNA, another nucleic acid. So the DNA, or pieces of it, are copied into RNA molecules, and that happens inside the nucleus. And another thing about the nucleus is there's usually a dark staining region somewhere in the nucleus, or maybe several locations in the nucleus. Those are called the nucleolus, or the nucleoli, if there are several of them. The nucleolus is the site where ribosomal pieces are put together. The ribosomes act out in the cytosol, but the nucleolus is the site where ribosomal RNA is made and where the ribosomal proteins are put together so that you can make the ribosomes here in the nucleolus. Those ribosomal parts have to leave the nucleus to go out into the cytosol to do their jobs. But those are kind of the big things about the nucleus. Ribosomes are the next structure we want to focus on. Ribosomes are the site of protein synthesis. That's their job. They're involved in making proteins. Remember that proteins are chains of amino acids that fold up on themselves and do jobs like they're enzymes, they're structural, they're transporters, they do all sorts of things inside of our cells. Ribosomes are the site where these proteins are made. Ribosomes are made of a large and a small subunit. Each subunit is made up of ribosomal RNA, so some nucleic acid, plus some proteins. Okay, the large subunit is made up of ribosomal RNA plus proteins, so is the small unit. Different proteins, different ribosomal RNA. And each of these subunits are put together in the nucleolus of a nucleus. Now, when we look at the cell, where are ribosomes doing their job? Well, they're doing their job outside of the nucleus. So they have to leave the nucleus through those nuclear pores, and then they do their job of making proteins in one of two places. There are two different types of ribosomes. We call them free and bound. Free ribosomes and bound ribosomes. So if we're looking at a cell, here's the nucleus, that's where the ribosomal subunits are made, and then they're shipped out of the cell. Free ribosomes, I'm going to draw them real tiny because they are very, very small structures, are in the cytosol. They do their job in the cytosol. They are not connected to any other organelle. And they help to make proteins that stick around, that function in the cell. Okay, so the proteins that they make tend to function inside the cell. There are other ribosomes that structurally are very similar to the free ribosomes, but those ribosomes become attached to a structure called the endoplasmic reticulum. And so they become, and I'm just going to draw little dots on my endoplasmic reticulum. Just write down ER for endoplasmic reticulum, but they become bound to the ER. So I'm going to write down attached to endoplasmic reticulum. Look ahead if you want to learn how that's spelled. And these ribosomes make proteins that tend to function outside of the cell. Okay, so the proteins made by these ribosomes tend to be secreted by the cell or put into vesicles. So they tend to have different jobs, but both bound and free ribosomes are doing the job of translating RNA into proteins, of making the proteins. All right, that's it for this video. Oh, wait, one quick question. I have some response questions scattered throughout this video. The response question here goes something like this. Many antibiotics target and disable bacterial ribosomes. Why does this lead to the extermination of harmful bacteria? I'm not going to address this question, but look for an extra credit assignment that you can earn some extra points on by thinking about these response questions that we see through the next couple of videos. So look for the assignment, the extra credit assignment. All right, that's it for now.